Okay, thank you very much. Well, you already heard my name in the title, so let's get into it. It's not really a phylogenomics talk, so I'm sorry if that's what you're here for, but hopefully it's still interesting. So just to give a little bit of background on Mendelian randomization, or MR for short. So MR is a sort of a class of methods that uses genetic so-called instruments, often SNPs, so you can just think of them as SNPs, to figure out whether one phenotype, which is often called the exposure trait, affects another phenotype, which is often called the outcome trait. And the reason a lot of people are interested in MR is that this is, under certain assumptions, a causal effect estimate, in the sense that this is true, it can provide valid estimates regardless of confounders of the exposure-outcome association. And so I've illustrated this in this little DAG graph here. So X there are the genetic instruments, or SNPs. E is the exposure phenotype, and O is the outcome phenotype. And U is a potential unobserved confounding trait, right? And so we all know correlation does not necessarily imply causation. And so E and O might be correlated, even though there's no causal relationship, just because of this unobserved confounder. So under certain assumptions, you can get a valid estimate of this effect of the exposure on the outcome. And just to sort of illustrate in practice a simple version of what that looks like, there's something called the ratio estimator for a single SNP. And so now I've kind of annotated this DAG with edge weights. So this beta XE and beta XO, those are the effect size estimates for this SNP on the exposure and outcome phenotypes that you would get from, say, a GWAS. And we want to estimate beta EO, right? And so under the assumption that the SNP does not affect the outcome trait except through the exposure, beta XO is just going to be beta XE times beta EO. And so just doing a little bit of simple algebra, you can see that beta EO is going to be equal to beta XO divided by beta XE, right? So this is just a very simple estimator for the effect of E on O. If you have multiple SNPs, there's a number of different methods you can do, like inverse variance weighting and two-stage least squares, but this is just sort of the basic idea of MR. And so I've alluded to these assumptions and already named one of them. There's a few assumptions underlying MR. One is that the instruments affect the exposure. Two is that the instrument is independent of confounders of the exposure-outcome relationship. And three is that the instrument does not affect the outcome trait except through its effect on the exposure. And in practice, there's several reasons why these may be violated, and often are. One of those is population stratification. Many of you are probably already familiar with this, but just to introduce the concept, this is a pervasive problem in GWAS and also MR. So SNPs may be spuriously associated with a phenotype, even if they don't cause the phenotype, due to differences in allele frequencies in subpopulations included in the study. And so I just have a basic illustration of that here. This is a BMI GWAS run in two different mouse strains. And I have two Manhattan plots basically showing the effect sizes of the SNPs. And the one on the top is with correction for population stratification, I believe using a linear mixed model. And on the bottom is one without correction for population stratification. And you can see, there's significant SNPs all over the place, which is pretty implausible. And the reason for that is, you can have two different strains that may be different in BMI, and there are some SNPs that cause that. But if you don't correct for population stratification, what's going to happen when you run your GWAS is basically every SNP where there's a different allele frequency between the two strains is going to show up as associated with BMI, right? So you're just going to get a lot of false positives. And so going back to the assumptions I listed earlier, now I've added population stratification to that graph. That's Z there. So that your population structure affects the distribution of the genotypes. It also affects it can affect the outcome trait as well as the exposure trait. as well as the unobserved confounder, so it can violate these assumptions. So some people believe population stratification is no longer really an issue because we have things like PCA and LMMs to correct for this, but it's been shown that even after applying these, there's still some residual stratification, and when you have these really big biobank data sets, even that small amount of residual stratification can be enough to cause false positives. But even if you don't believe that, there's other things that can cause problems like cross-trait assortative mating, weakened instrument bias, which I won't have time to talk about, but our method is also robust too. Another big issue is pleiotropy, which is something we don't cover. Okay, so now getting to what we actually do. Our method is called MR-Twin, and it's based on kind of a simple key idea. And throughout the talk, I'm going to use an example of if we have trio data. So suppose we have genotypes from both parents and also their offspring. Although we also our method also works if you only have one parent or if you have sibling data. And so the key idea is that the genotypes of the offspring are independent of population structure given the genotypes of the parents, right? So if I have your parents' genotypes, I know the probability that you inherited your genotypes from your parents. It doesn't give any additional information to know your ancestral background, right? And so now I've added that to the graph there. So I'm now denoting A as the parental genotypes, and now X are the offspring genotypes. And you can kind of see there, A is sort of blocking this backdoor path from the offspring genotypes to the confounder. And I talked about population stratification, but now this works for several types of confounders. And so our method is based on this principle is provably robust to these types of biases. And I just want to emphasize, I talked earlier about effect size estimation, but here we're focusing on a hypothesis test for whether the exposure phenotype has a causal effect on the outcome phenotype. Okay, so that's sort of the basic key idea. Here I have a schematic of our method. On the left are some graphs I've already shown you. On the right is a little drawing I did. So here we have two parents, and I've drawn their two little chromosomes. And then we also have the offspring genotypes. And you can see it's a mosaic of the genotypes of the parents. And so where the name MR-Twin comes from, based on an idea from Bates et al. Called the digital twin test, is what we do is we simulate so-called digital twins from these parents. And we have the parental genotypes, so you can imagine we can just simulate meiosis and recombination and generate different mosaics of the parental genotypes. And then we can compute a statistic, which I'll kind of overview later, for both the true offspring and for each of these digital twins. And then the MR-Twin p-value is the quantile of the true child's statistic among this set of digital twin statistics. So if you're familiar with permutation tests, it's a similar concept, except here it's just conditioned on the parental genotypes, kind of generating a null distribution of genotypes. So now the two parts of that. The first step is sampling conditioned on the parents. We want to sample digital twins, the genotypes I'm denoting as X tilde, in a distributionally equivalent way to the true offspring genotypes. And normally this is, can involve some challenges like, you have to do haplotype phasing and then simulate recombination with an HMM or something. But one of the nice things about Mendelian randomization is usually the way it's done is you run a GWAS for the exposure trait, and then you pick a certain tag SNP for independent loci across the genome. So you have this set of independent SNPs, and then we don't have to worry about all this phasing and recombination and stuff. We can actually just do a simple Bernoulli draw from the parental genotypes. So if a parent has a 2 genotype, you're definitely getting a 1. If a parent has a 1, you have a 50-50 odds of getting a 0, 1, and so on. So it's simple and fast. For the statistic, I've written it in there, but don't worry too much about the notation. The basic idea is that we want to predict the outcome trait of the true offspring using either the true offspring's genotypes or these digital twin genotypes. So if the genotypes have an effect on the outcome via the exposure trait, then the true offspring genotypes should predict the true offspring outcome better than these digital twin genotypes, which are randomly sampled from the parents, right? If there's no effect, then it should make no difference whether it's the true offspring's genotypes or these ones we've sampled, and that's kind of what our statistic is doing. Okay, so that's kind of the basic high-level overview of our method. Now I'm going to talk a little bit about our results. So sorry for this heavy text slide, but basically just showing, just describing our simulations. So for a variety of settings, we simulated 50 to 100,000 individuals, split evenly between two populations using Baldwin-Nichols model. We call this the external data that we run. You run standard MR methods on. We also simulated 1,000 trios, which we call the trio data, that we can run our method on. And this is designed to be sort of typical of the sort of data set sizes you'd see in reality. Then we simulated our exposure and outcome phenotypes. For each of those, we simulated 100 causal SNPs, and we had a polygenic model with the heritability equal to 20%. And I guess the key point of this is generating population stratification. So we have a binary label, right, for each individual denoting which population they're in. And we allow this label to have an effect on the exposure trait and the outcome trait. So this kind of generates this effect from genotype to outcome trait through the population structure. So that's how we have stratification. We then ran a GWAS to generate summary statistics for the external data. And then we ran some standard MR methods. If you've worked in MR at all, you're probably familiar with these on the external data. Then ran MR twin and another family-based method from Brumpton et al. On the trio samples. Okay, so I've been talking a lot about this. Let's see the results. This is kind of the key result. So on the left here, I have the false positive rate of these methods on the y-axis, and the x-axis is increasing confounding effect size. And you can see that these standard MR methods are highly miscalibrated as the confounding effect increases, which is expected because this is not a type of confounding they're built to deal with, but illustrates the point. And you can see the family-based methods are much closer to calibrated. MR twin is the blue line, and Brumpton, this filtered versus unfiltered has to do with weak instrument bias, and that's why you see a little bit of inflation. MR twin's also robust to weak instrument bias. On the right, I have power on the y-axis and increasing causal effect size on the x-axis, and this is sort of where the standard MR methods get their revenge, right? They're much more powerful, and there's a couple of reasons why this could be. First, obviously, they're using much larger sample sizes, right? Like 100,000 versus 1,000. Another is there could just be some problem with our method, limitation with our method, and to assess that, we reran these methods just on the trio data, so now everything has the same sample size, and lo and behold, right, the standard MR methods are still have an inflated false positive rate, but now we can see the power of the methods is much more comparable. And again, MR twin's the blue line, and it's similar to or more powerful than most of the MR methods. And just to sort of, I'll just briefly talk about our real data application to the UK Biobank. You can isolate about 1,000 white British trios in the UK Biobank, and of course, something like 300,000 unrelated white British individuals. We ran the MR methods on these, and recapitulated a lot of the same trends we saw in our data. So I have a couple trait pairs that are definitely true positives, like LDL cholesterol affects total cholesterol, a couple trait pairs that are more unclear, and then a couple of implausible ones. And basically the MR methods largely get what you'd expect. The standard MR methods, whether because of sample size or confounding, find a lot more significant trait pairs. Okay. So in summary, MR twin is a test for MR effects that uses a conditional randomization approach to provably avoid false positives from population stratification, and I didn't have time to talk about these, but also cross-trait assortative mating, dynastic effects, and weak instrument bias when using trio data. Some limitations currently, as I alluded to before, power is limited by small currently available family sample sizes, especially trio sample sizes. And trio data is really where this works best. So hopefully that will be improved in the future. Hopefully there's just more trio data coming. I didn't have time to talk about these, but there's also duo and sibling modes. They don't fully avoid confounding, however, because we don't have the actual parental genotypes. We have to infer them or estimate them. Also, MR twin takes longer to run than existing methods, but it's still plenty fast. I mean, we're talking about like a couple minutes versus a few seconds in our UK Biobank analysis. In the future, we'd like to improve the sibling mode and extend to further pedigrees, like if you have cousins or second cousins or something. And it would be nice to be able to incorporate correlated instruments, because it could help increase power, but then you have to do all this phasing and stuff. So, but that could be of interest. And I'd just like to acknowledge my co-authors. Boyang was my co-first author on this paper, Stephen, who's a great undergrad, and Eleazar and Sriram, who co-supervised this project. I'd also like to thank my lab mates, and here's our paper information, and we also have a GitHub. And thank you very much.